Welcome. It's my pleasure to introduce the Basic Science Wrap-Up. This year, there were almost 200 abstracts presented in the Basic Science section, and these covered a wide array of topics from gut microbiota and liver disease to liver immunology, fibrosis and stellate cell biology, liver development and regeneration, and more. So let's jump right into a microbiota presentation, one that was given by Marissa Isabel Keller from the Pierre Bohr Group. They performed a meta-study of almost 17,000 fecal metagenomes from 129 different studies, covering healthy subjects and 32 different diseases. They developed an enterotype dysbiosis score using something they called fuzzy clustering, based on classification strength to quantify the level of dysbiosis in patients with liver disease. This score was increased in patients approaching ACLF and predicted survival probability, and increased in patients approaching death. If you're interested in the enterotype dysbiosis score, you should know that they've created an online tool for this, which you can find at this link. Another interesting microbiome talk was given by Wenyi Gu on behalf of MicroPredict and Predict Study Group, and the study group of the EASL Cliff Consortium. They proposed that bacterial translocation caused by leaky gut syndrome could play a role in condition worsening in acute decompensation or acute on chronic liver failure, and this would drive bacterial infection. So the study includes 93 patients from the Predict cohort, and focuses on those with decompensated cirrhosis, classified into subgroups. They sampled 11 different anatomical locations and did 16S metagenomics. So the authors performed many exhaustive analyses, for example, looking at similarity in longitudinal samples, and found that patients who developed ACLF had increased dissimilarity, which they ascribed to bacterial translocation. Another exciting abstract was presented by Johan Krag Hansen from the University of Southern Denmark and Odense University Hospital in Denmark. The PIs were Alexander Krag and Torben Hansen. They integrated data from 10 omics profiles, including microbiome, lipidomics, metabolomics, genomics, and transcriptomics, to profile different stages of liver disease, and they ultimately included over 10,000 features. Over 50% of those features could be associated with liver disease severity, and they could be hierarchically clustered into 6 patterns of behavior across worsening disease stages. So there's lots of interesting biomarkers and mechanisms to dig into there. Now we can jump into an immunology topic. So Valentina Venzin from Matteo Iannacone's lab at Vita Salute San Raffaele University in Milan, Italy, investigated CD8 T-cell dysfunction in the context of hepatitis B virus infection, or HBV. The authors ultimately propose a model in which CD4 T cells can enhance the function of Kupffer cells, or license Kupffer cells, to produce interleukin-27, and in so doing, restore the cytolytic function of CD8 T cells. So the authors used multiple animal models, including an HBV transgenic mouse model and adoptive transfer, to show that co-transplantation of CD4 T cells increases the expression of interferon gamma and reduces the viral load in mice. Using live imaging, they demonstrate that CD4 and CD8 T cells interact via Kupffer cells, and that the Kupffer cell phenotype is improved by CD4 T cell presence. Importantly, they demonstrate that Kupffer cells are essential for mediating the positive effect of CD4 T cells, and if they ablate Kupffer cells, the CD4 T cells can no longer improve the CD8 phenotype. The interaction of bile acids with NK cells was also described by Ahmed Salhab from Hadassah Hebrew University Hospital in Israel, supervised by Johnny Amer and Rifat Safadi. They report that NTCP is expressed in NK cells upon fibrosis, and that its expression increases with fibrosis stage. The NTCP expressing NK cells could take up taurocolic acid, and the authors further show that there is a function for STAT3 signaling in this process. With regards to fibrosis and stellate cell biology, Mathieu Bougon, supervised by Jérôme Ferret and Nathalie Thierret at the University de Rennes, France, developed a rule-based model using kappa to simulate the dynamics of hepatic stellate cells and CASIM for simulations. The stellate cells were modeled as agents with 7 physiological states, with interactions with TGF-beta1 molecules and an impact on collagen-1 levels. Their model predictions were validated using experimental data. Ultimately, the authors conclude that an inactivation loop of hepatic stellate cells is critical for the elimination of activated hepatic stellate cells during fibrosis reversion, and that reactivation loops of inactivated hepatic stellate cells are necessary to sustain fibrosis. An accumulation of the inactivated hepatic stellate cells during chronic liver disease was predicted by the model and confirmed using RNA sequencing data, identifying these IHSCs as novel markers of fibrosis progression. So the manuscript is on bioRxiv and it's been accepted in PLOS Computational Biology. R.T. Sharma from the Institute of Liver and Biliary Sciences in India presented work on lymphatic endothelial cells. They demonstrate an expansion of the lymphatic endothelial cell network upon partial hepatectomy and wanted to investigate the function of the secretome. They sorted lymphatic endothelial cells for mass spectrometry and identified numerous enriched secreted proteins, including relin, which they speculated could activate hepatic progenitor cell proliferation. So they grew hepatic progenitor cells as organoids and co-cultured these with the secretome from lymphatic endothelial cells and showed that this could increase proliferation, which they ascribed to relin activity. Alexander Walker from the Luke Bolter lab in the UK presented their work linking collagen integrin signaling to bile duct morphogenesis. They found that integrin alpha-2 is upregulated in specific regions of ductular regeneration. They profiled integrin alpha-2 expressing cholangiocytes with RNA sequencing and showed that integrin alpha-2 knockout or FAC inhibition could reduce cholangiocyte migration, suggesting that these otherwise can mediate an EMT-like transformation, which you can see here. Finally, Rita Manco from Isabelle Leclerc's lab at UC Louvain in Belgium presented their work in the general session. In chronic liver damage, the persistent injury and inflammation leads to proliferation of ductular cells known as ductular reaction. The authors propose a model in which the fate of the ductular reaction cells is determined by their endothelial cell environment. They used 6 weeks of carbon tetrachloride to induce liver injury and ductular reaction. They used spatial transcriptomics to differentiate between ductular reaction cells that are embedded in laminin, here, versus those that are not, and assessing their gene expression profiles, showing that laminin-embedded ductular reaction cells were more cholangiocyte-like. Next, cholangiocytes were cultured in a double spheroid model with varying compositions of ECM to study the impact of laminin on cholangiocyte behavior, showing that laminin promotes a biliary phenotype. Interestingly, liver damage induced the emergence of CD34-positive liver sinusoidal endothelial cells, and this is a marker that demonstrates that these endothelial cells are becoming capillarized. The authors show that these capillarized liver sinusoidal endothelial cells have a pro-fibrotic phenotype and impede the differentiation of ductular reaction cells into new hepatocytes during chronic liver disease. They also wanted to test preventive strategies, and to do they used an inhibitor known as BAP602770, which is a soluble guanylyl cyclase activator that has an antifibrotic effect. So when they apply this activator, they were able to inhibit capillarization, and following the ductular reaction, they show that this leads to transdifferentiation of the ductular reaction into hepatocytes. So preventing liver sinusoidal endothelial cell capillarization can influence ductular reaction and act as a form of therapy. So that was the wrap-up. Apologies again to the many excellent presentations that I could not include, and I encourage you to watch the scientific sessions on demand to catch up with or watch again your favorite talks. See you next year in Amsterdam.